We mimic the effects of crossing over. Okay? Now we take this and do the test cross on it. Combine this with your homozygous recessive. So we do the top of the left and the bottom of the right. So the top of the left and the bottom of the right will give us this one. Can you see that? The top of that one and the bottom of that one gives us this. Okay. And the bottom of this one and the top of that one gives you this. So that's how you know how to draw what the recombinant combinations are going to look like. How do you know what percent they are? You don't. Someone has to grow a whole bunch of pea plants or fruit flies or something that produces a prodigious number of offspring in order to elucidate that. So the little drill that you'll become very proficient at just running through this, and it's always the same. Once you just learn how to go through this, it's always the same thing. And so, top of the left, bottom of the right, bottom of the left, top of the right, gives you those two. Then you take your F1, flip one side, like I did over here, top of the left, bottom of the right, bottom of the left, top of the right. And that'll give you the recombinant ones. So it'll just be the same in every situation. So, they've done a lot of work with fruit flies. You know these bug stations we see at the border? The Mediterranean fruit flies drove people in California. They lost billions of dollars in agricultural profits from this. These fruit flies can reproduce like nothing you ever saw, and their little larvae do terrific damage to certain types of fruit trees, which if you've ever driven down I-5 in Southern California, there's hundreds of miles of this beautiful agriculture down there. My dad told me one time that California produces more agriculture than almost any other country. And so they're on the look. They've learned a lot about fruit fly genetics, and fruit flies have the advantage of producing huge amounts of offspring. So you can count the offspring. Some graduate student takes a couple of flies and mates them, and then you get this hugest number of eggs. Takes care of the eggs, and so that they do well. And all the little thousands of eggs hatch into these little maggots. Okay, takes care of the maggots, gives them something to eat, so that they grow up nice and healthy. And then they go into a pupa stage, and the pupa stage then hatches into the adult flies. And then after all that, he picks the flies out one by one out of this maybe 30,000 fruit flies and notes the phenotype of each one of those fruit flies and writes it down in a notebook or computer or something. Now you may want to do that, but me, I'll pass. So they use kind of interesting notation for their alleles. The type of notation they use, we're looking at two different traits. We're looking at body color and eye color. Okay, and there's quite a bit of variation in these fruit flies. Why are we looking at fruit flies? Because you can't look at humans. Okay, this is going on in humans, but humans don't have enough offspring to be able to count the results and have them be statistically meaningful. So, it's kind of hard to keep the symbols straight at first, but you get used to them. The symbol that they use is an abbreviation for the recessive phenotype. So the recessive phenotype is ebony body. So we use EB But if it's got a plus by it, it's the dominant phenotype. So yellow body is dominant to ebony body, which doesn't have a plus by it. Red eyes, and they use, is dominant to maroon eyes, see? And so they use a symbol for maroon eyes, but if it's got a plus, it's the dominant form, which is red eyes. So what they did is they took an ebony body, red-eyed, and they had to say, since this is the dominant phenotype, they had to say it was homozygous, was mated with a yellow body, homozygous, maroon-eyed fly. And so what we're doing is making an F1 generation, basically. And you're only going to get one kind of offspring in the F1 generation. They were all yellow body, red eyes. Then the F1 was test crossed. The ebony body and maroon eyes gene are 20 map units apart on the same chromosome. So what is a map unit? You will see. This is how map units were created. If you've ever looked at a chromosome map before, the genes are, except one way of calculating where they are on the chromosome is by map units. Okay? And give the genotypes for participating individuals. Okay? Give the phenotypes of the progeny. Give the proportions for the progeny combinations and label them. So, here are symbols, and I already forgot what the problem said. Okay? But we're taking, it looks like, an ebony body and red eye. And we remember they said red eye homozygous? Because it could be they're homozygous or heterozygous. So we're taking ebony body, red eye, and what's the other body color? Yellow body, and that's the dominant one, maroon eyes. Okay? And so top of the left and the bottom of the right gives you that. And the bottom of the left, top of the right also gives you that. Okay? So this is our F1 genotype. And you have to go through all this to figure out what the F1 genotype is, because it's impossible to know whether the dominant alleles are on the same chromosome. Here they're not, are they? So we're going to do a test cross on this. And we're always going to do a test cross on the F1 generation. That's the drill. You're just going to learn this sequence of events. Okay, so there's our F1 generation, and we wrote it again over here. Okay, and so we're doing a test cross, crossing it with homozygous recessive. And so, top of the left, bottom of the right gives you that one. Bottom of the left, top of the right gives you that one. How do you figure out what the other two are going to be? You go back to this one and flip the right-hand side. And then do your test cross again. And so, I didn't bring my eraser over here. Blast it. Okay, and so we take, thanks, and redraw our F1. We'll keep the left-hand side the same, okay? And we'll flip the right-hand side. Okay, and then we take this and cross it with that one, the homozygous recessive. And so, top of the left, bottom of the right gives us that. And bottom of the left, top of the right gives us that one. How do we know what the proportions are? Here's where this map unit thing comes in. they're 20 map units apart. And so the total percent of the two you're gonna get the same amount of each one of these. Okay? And they gotta add up to 20. The map units tells you the total percent of recombinant offspring. The total percent is 20%. Okay? They're 20 map units apart. The more map units apart they are, the greater the percent of recombinant offspring that you'll get. Okay? And so, knowing that adds up to 20, these two gotta be equal to each other, and they gotta add up to 80, don't they? So that you get 100. So that's the whole thing. That's all. Now we're just gonna look at a few different twists, but you always go through the same thing. So in fruit flies, the allele for normal wing shape is dominant. Have you ever seen a fly with shrunken, teensy little wings? None of my students ever look at flies. I have. Does that say something about me? I'm not sure I want to know. But vestigial wings are wings that kind of never really form. And they must not work. And so those flies don't live very long. But long enough to be counted. The second gene is the gene for eye color. Okay. In this case, the gene for burnt orange eyes is recessive to the allele for red eyes. These two genes are 9.9 map units apart on the same chromosome. So, what progeny of genotypes and phenotypes and in what proportion would you expect when a female fly, heterozygous red eyes and heterozygous for normal wings, with both dominant alleles on the same chromosome, is mated to a fully homozygous recessive male? So it sounds to me like they're sort of starting you off with the F1. This is going to be a shorter problem. Okay? Because if they're mating something with the homozygous recessive, they're doing a test cross already. Okay? So you're already got what amounts to an F1 generation. So, we do the top of the left, bottom of the right, and that gives you that one. Bottom of the left, top of the right gives you that one. Then you take this and flip it and cross it with that, and you'll find that you get those two. Okay? And they were 9.9 map units apart, so the 2%'s got to add up to 9.9. With me so far? Okay. And so the other two, whatever 9.9 is subtracted from 100 is 9.9 is going to be 90.1, isn't it? So you get 45.05. You see how all that happened?
Yep.
It's the same drill. And so then you'll be asked, and you'll be doing this on my quiz next week. Only you'll probably be asked to get the F1. And you'll be asked to write the phenotypes of the offspring too. So you see that's not burnt orange because it's got a dominant gene, which is red. And that's not a vestigial wing because it's got a dominant one, so it's got a normal wing. Here we have burnt orange eyes and a vestigial wing. Here we have red eye and vestigial wing. Here we have burnt orange eye and normal wing. And you'll get used to those symbols after a while. It takes some getting used to. Okay? And so round and round we go. For the same type of fruit fly as in the problem above, the allele for gray body is dominant to the allele for black body, and the allele for straight wings is dominant to the allele for curved wings. When a black body curved wing flies were mated with a gray body homozygous straight winged, the F1 flies were all gray body and straight winged. And so this time we're given numbers that the graduate student actually counted. Look at those offspring. How would you like to count those? And so, okay, we're doing getting the F1. And so this would be your two recessive traits. And I forget even what they were, probably black body, curved wing would be my guess. Yeah, I think that's right. And this is gray body, normal wing. And so top of the left, bottom of the right gives you that. Bottom of the left, top of the right also gives you that. And that's your F1 generation. What are we going to do with it? We are going to test cross. Good response. So we redraw it over here, and we do do our test cross. F1. Some people call it a back cross. It's the same thing as a test cross. Homozygous recessive. Top of the left, bottom of the right gives you that. Bottom of the left, top of the right gives you that. Then you take this, draw it off to the side, and flip one side. And do the same. Top of the left, bottom of the right, bottom of the left, top of the right gives you those two. Now, we're stuck with these numbers. And I think you're asked for how many map units apart they are. Okay? Or some such thing. I forgot. But all you got to do, you guys remember how to do percents? You add these all up, and whatever the total is, so you find your total, and then you add those two up, and that will be divide by the total. And that'll give you a decimal fraction, which you can convert to percent by moving the decimal over 2. So they've done that here. These are your recombinant offspring, and there's your total. And so they're 22.7 map units apart. See? And there we show a little map, 22.7 map units apart for the body color and the curved wing. Okay? Now what I'm going to do is work some problems, one of each type. And I'd hang around, boy, because these are going to be very similar to what's going to be on the quiz. And these are the problems I handed out to you. Okay. And of course, I forgot my eraser. Thank you. So, oh yeah, I should bring my. Could you bring all my? So let's do them.
I'll go there.
So, in humans, the allele for dark hair is dominant to the allele for light hair. So let's give that a symbol of H. Okay? I'm doing number 2. Number 1 is easy. It's, yeah, for each of them. Give the number of gamete genotypes. Remember the 2 to the n rule? It's the number of heterozygous ones you got.
Are they all
They're all
8?
8, yeah.
And so let's see. The first one is going to be A.
And then 8.
And the second one's going to be 8.
The last one's 8.
And the third one is going to be 8. Yeah.
And then write them too.
And then draw them all out. Yeah.
See, I only get.
So there'll be one like that.
You draw one of them out.
Draw one of them out. Oh my goodness.
Can I get one of those sheets?
Yeah, this is my last
That's it,
one.
okay, no.
So you just got to start grinding them out. I may not do them all, but so there's going to be one like that. Okay. And you just go through and see. See what I mean?
Yeah.
Yeah.
So
Okay.
we're supposed to have
Got it.
8
Thank
of.
you, ma'am.
So you know that you stop when you have 8 different
I
ones.
don't understand how you get 8 from that when you have. to-
Because there are 3 heterozygous pairs there.
Oh, I know.
So 2 to the 3rd. 2 times 2 times 2.
Can you write out all 8?
Can I write out all 8? Okay. Uh,
Yeah, all of them are 8, 8, 8.
They're only,
They're all 8.
you only count, you only count the heterozygous pairs, and then there's 3 in each of them. Because 2 capital Ds is homozygous.
Because
and
you have your P,
so
Q,
then,
and T.
You're right.
So what am I missing?
1, 2, 3, 4, 5. So let's see here. I think that's different. So there's 6. You really want all 8?
Well, because I only came up with 4 and I couldn't figure out how you got the other 4.
Well, I must have a bunch of them here that you don't have.
1, 2, 3, 4,
Let's
5,
see.
6. I have all.
You've got 8?
Yeah, F, G, lowercase h, capital J, you have that one. F, G, capital H, lowercase j, you got that one. Well, you do them all capital X, and then all you have to do is copy them again with lowercase f and get you.
Yeah, there's,
Just do all the capital F's first, and then copy
yeah,
the
then you
same
just
thing with
do
lowercase
start, do all the capitals first, and then do one little one,
f.
and move the little one to all the different positions, and then, you know,
1, 2, 3.
you gotta have a sort of a system. I was just zapping them out. I think all these are different though,
Yeah.
but get the idea? This isn't
No, there's
real hard,
duplicates. Oh, no.
just a matter of taking the time. Okay, so, got the idea? And you quit when you have 8 different ones. Okay. Okay with that one? There will be one of those. So in humans, the allele for dark hair is dominant to the allele for light hair. So we got a light haired man, and so I'm going to use little h. Okay? And we know that he's got to be homozygous because that's the recessive. You know, this is the man's hair color, and he has blood type O. So that's the man. Okay? And a light-haired, okay, his wife has dark-haired and blood type AB. So the question is, if with dark hair, is it going to be homozygous or heterozygous? But it tells you her father has light hair, so you know he had to be heterozygous. She did. Okay? Because that one came from the light-haired dad. And the blood type was AB. How many different genotypes? Remember, the gamete's only going to have one allele of each kind. How many different gamete genotypes can you get out of this? One. It's going to be HO. That's it. How many different gamete genotypes can you get out of that one?
4.
4. You get the big H with the big A, the big H with the B, the little h with the big A, and the little h with the B. Okay? And so, what is the probability they will have a child with dark hair and blood type A. Well, the next thing we gotta do is do our Punnett square. Okay? So, and there will be one like this on the quiz. And you'll have to figure out, I mean, it may not just be one row of boxes, could have 2 rows of boxes, or could have, I doubt I'll have you do one with 16, because it's too time consuming. But, and so here's our male over there, and here are the 4 gamete genotypes for the female. And so then we combine these and we get so what are the phenotypes for these 4 boxes? Well, we got dark hair blood type A, dark hair blood type B, light hair blood type A, and light hair blood type B. What is the probability they will have a child with dark hair and blood type A?
25%.
25%, 1 box out of 4. Oops, type A. What is the probability they'll have a light-haired child with blood type B? Well, that'd be that one, 25%. How many different phenotypes could their children show? 4. Or all would be different phenotypes, wouldn't they? So there'll be one like that.
Okay.
I did a bunch of doodling on here. On the back side, I kind of worked problem 4, but first we're going to do problem 3. You will see a problem just like this problem 3, and so it asks you, gives you, it tells you something about the child and the parents, and says, could these parents have had this child? Could these parents have had this child? And you can't just say yes or no if you want to get full credit. You've got to give some kind of substantiation. So item A, if you look at what the child has, blood type B, blood type M, and Rh negative, notice that the father had MN. And so what you're going to have to do here, A, B, you're going to have to do 3 Punnett squares.
Okay.
For A, I'm afraid, for full credit. If you show the 3 proper Punnett squares, you're going to have to do 1 for, okay, we have A and AB. Now the question is, I would call it AO, or you're going to have to do a lot of Punnett squares if you assume, I think AO gives you more possibilities of different, rather than AA. See what I'm saying? So if you call it AO, and the father's AB, then we're going to go AB, AB, BO, B-O. So that would work.
Father
It's
Father is
AB.
AB.
Yeah.
The what?
Father's
Father's
AB,
AB.
so it should be
Oh,
AB.
did I write it down? Oh, the mother, the fa- well, I know the father's oh, yeah. I wrote it wrong. Don't do that.
The next one's AB. I don't know.
Okay,
How
so
come you would've put BB? Why wouldn't you
So-
put BO?
just let me do it again.
Because the father says AB.
Yeah.
Why no, but-
I wrote it in there wrong.
It just meant, he meant
So-
AB, he just accidentally put BB. So for A, we have
So that would work for that one, for the child, because the child's got blood type B, and that would be that, wouldn't it? If you didn't start with AO though, it might not have worked, I don't know. But it's better to, you get more possibilities if you use AO and BO. So the next one is the M and the N thing. And I would, those are blood antigens as well. I would use MO and MN. So then we'll get MM and MN and MO and NO. My goodness. And so that would work. That would be that one there. So that's possible. And finally, the father's Rh positive, and assume that it's RHO. Little, best to call it little r probably. And so that's the father's Rh positive and the mother's Rh negative. And so and the child is Rh negative, so that works, doesn't it? So yes, it's possible. And if you showed all three of those Punnett squares and just said yes, that would be a great answer.
On the Punnett square, on the second one, you said MO was the one, couldn't MM also be?
Oh yeah, it could. Yeah,
On the, instead of MO.
those are both type M.
Yeah.
Absolutely, Cassandra. Okay. So the next one, you gotta go through, I think, the whole thing again. Want me to do that?
Yeah.
Okay. So, and we'll call it AO for the father, and OO. That ought to come out easily. So,